I want to welcome everybody to the last, but also the best session of the whole day's events here. And I want to thank Josh Deney for putting together a fabulous program, but in particularly for naming this last session, What If? And I think that really captures the incredible future that we have thinking about these new techniques. You've heard about fabulous new applications and cures all day, but the future is bright. And we have to remember that in these difficult days, that the science is going to get us through, and it really is going to be a bright future once we get through the cloudy bits. So without further ado, I want to introduce our first speaker today, Yuyan Cheng from the Department of Ophthalmology, who's going to be talking about some single cell and spatial transcriptomics.
All right, thank you, Claire, for the introduction. Thank you to Josh and Benny who invited me to speak here. And thank you to everyone to stay towards the end. I guarantee it's gonna be one of the best sessions. And my talk today is going to describe some of the working in progress since I started my lab last year. And then the title will be, What If Single Cell and Spatial Transcriptomics Can Decipher the Complexity of Neurodegenerative Vision Disorders with a primary focus on glaucoma. So we know that many retina and optic nerve neurodegenerative disorders, including glaucoma and many other optic neuropathies, are highly complex due to involvement of multiple cell types from RGCs, their neighboring cells such as amacrine cells and glia cells or blood-borne immune cells that's like distributed across the retinal layers and in the optic nerve. And so the behavior and the state of the cells are essentially, if we zoom in into each cell, they are decided by a cascade of molecular events occurring during disease onset to disease progression. So currently there's a lacking of a comprehensive understanding of this multi-layered, multicellular, multi-molecular complexity, and this actually creates a roadblock for us to develop efficient or accurate diagnosis and effective treatment to either halt the disease progression or restore visual functions. So the advances in single-cell and spatial transcriptomics are really transforming our way to understanding the biology of a cell or investigating complex neurodegenerative disorders, including vision disorders. So these technologies, as many of you know, allow us to explore each cell as an individual unit, uncovering the heterogeneity that underlies disease onset and progression. So for example, the single-cell RNA-seq, with single-cell RNA-seq, we literally just break apart the tissue to release to dissociate two single cells, and each cell is going to get a unique barcode. And within that cell, each RNA transcript is gonna get additional transcript. So that means like each cell and its message in that cell, all of them are barcoded, so you know where they come from and which cell they come from. And spatial transcriptomics, on the other hand, preserves tissue architecture by measuring RNA directly on the tissue section. And because it's direct to imaging, you can actually integrate histology and morphology. And in this way, you actually get multiple layers of information from transcript abundance to their location, the tissue space, and morphology all in one place. So by combining both single-cell and spatial data, we can actually not only map the tissue architecture, how they are changing across time or like in disease progression, and we can also map the cell-cell communications in actual tissue space. And eventually, we can identify the molecular events or the drivers that drives the disease onset or progression. So this really helps us to pinpoint or like identify the key cellular and molecular players that's most critical for disease pathogenesis. And I want to point out the richness of and the scale of the data, often reaching millions of measurements, really opens the door to powerful computational modeling. So for example, people have been using machine learning, neural network, and even foundational models, which gathers like millions of single cell data, and to use them to predict genetic perturbations, biomarkers, or even to design diagnostics tools with high precision. So this integration of biology and AI is really transforming how we understand and treat complex disorders. So with that kind of framework in mind, we wanted to apply it to glaucoma, which is actually a perfect example, as it is a complex, multicellular, and multi-molecular disease that has no treatment at this stage. So I think you've heard enough about glaucoma in the last session, and it's actually a heterogeneous group of optic neuropathy with elevated intraocular pressure as the primary risk for glaucoma. And currently, even though the patients, they have IOP's relief, they still develop glaucoma and eventually, some of them eventually lose their vision. So that actually means that we need to dive deeper into the molecular and cellular events on what's going on when the IOP is still like elevated, and if we can come up with like better treatment for that. So with this in mind, we set up a collaborative team with glaucoma specialists in the Kirby Center, including Drs. Choi, who gave a really nice talk in the last session, and Drs. Ross and Schindler's lab. And we are trying to build a comprehensive single-cell and spatial atlas for in the mouse model of glaucoma, primarily focused on the mouse model with elevated intraocular pressure. So in this collaboration, we are trying to use the microbead injection model, which can consistently increase eye pressure across time to mimic the patients that has constant eye pressure in clinical settings. And after the eye pressure is elevated, we are collecting the retina and the optic nerve from the very early time points all the way to when their eye pressure goes back to normal. And one set of the mice are going through single nuclear RNA-seq, and then the other set of the mice are going through spatial transcriptomics. So this is the first set of mice we have collected so far, and we have four to five mice per time points after the IOP is elevated, and those mice are age and sex balanced to remove potential confounders. And we have three major groups over here with the red one, the bead injected, and then we only keep the ones that has high IOP over 21, which is considered which is considered to lead to glaucoma phenotypes in mice. And then we also have vehicle-injected mice together with mice that has no injections at all as a baseline control. And we take one mouse from each time point and trying to get a sense of how our single nuclear RNA-seq pipeline works. And we generated this data set with 50,000 cells. And as you can see here, with this 50,000 cells, we can identify major retina cells, including cones, rods, bipolar cells, amacrine cells with a lot of subtypes. And of course, the rods are major components of this data set, accounting for over 60% of all the cells, while the RGCs are a very small population, around like 1% of the whole data set. And on the left, I'm just showing you, this bubble plot is just showing you those cell types are expressing, those clusters are expressing their respective markers, which shows the good quality of this data. So this is still very small data set. As I said, it's a preliminary, and our goal is really to use cost-effective single nuclear RNA-seq technology to generate large-scale data up to like 500,000 cells for both retina and optic nerve. And only with that scale, we will be able to detect different RGC subtypes and also start doing something fun like AI, like machine learning modeling. But with that, with this data set, the first question that comes to our mind is that what are the cell types that are most responsive to bead injections, right? So to systematically analyze this, we use this, we come up with, we utilize this machine learning-based framework, which is called Augur, and it's a random forest-based classifier. So it's very intuitive. So basically how it works is that if you think about like a cell that is very like profoundly affected by a treatment, in this case, elevated ocular pressure, right? So it should naturally separate, like easily separatable from the control counterparts by just looking at the molecular signatures. So in other words, we can tell, we can predict without knowing the conditions whether the cell comes from a treated or untreated group by doing like predictions. So we try to train a classifier to separate the cells from treated or like bead injected or control groups, and then try to estimate the accuracy for this prediction, right? And then the more accurate the classifier is, that means the cells are more separable, meaning that they are more responsive to elevated ocular pressure. So with AOC, you have heard this like all the time, being like 0.5, which is random, meaning that the model does not predict any separation between the treated versus untreated. And then the higher the AUC is, the better the classifier is, and that means the cells are more responsive to a perturbation. So the figure is a little bit blurry for some reason, but here is what I'm showing you here is the AUC values for each of the cell type across different comparisons. So the comparisons are vehicle versus control. Just a reminder here, the vehicle is the one injected with balanced saline, just as a control for the bead injection. The control is baseline with no injections at all. And then the bead, so the comparisons are vehicle versus control, bead versus control, bead versus vehicle, and bead versus vehicle and control combined. So very like obviously you don't you don't expect to see like separations or like responses from each of the cell types to between vehicle and control, but you do see some separations or responses between bead versus all the other control injections for most of the cell types except for rod cells. So that means many cell types actually in the retina, they are responsive somehow to different extent to the elevated ocular pressure. And this you can further see if we separate the AUC out like among different cell types, that rod is least responsive to elevated ocular pressure, while the others are, including RGCs, are responsive to the bead injection to some extent. So we further look into the molecular level, right? So the idea of doing that cell perturbation analysis is trying to narrow down to the cell types that we are really focused on for like deeper downstream analysis. And then we further do differential gene analysis, trying to see which cell types are most responsive. And again, across the three methods, the well-established methods for doing differential gene analysis, we found two of them demonstrating that RGCs and in some of the methods, the bipolar cells being most responsive to bead injections. And again, the vehicle versus control, they don't seem to show much difference. So again, this is a preliminary data, and then we don't want to cherry picking some of the genes that we are interested in. We're just waiting to increase the data size so that we get like more confidence before we are really like narrowed down to like target cells or molecules. And once we do that, and our goal is trying to use spatial transcriptomics to validate some of the molecules that's interesting to us. And it's not just validation, it also helps us to understand the cell-cell interactions and how these important genes that are responsive to elevated ocular pressure in different cell types, how they are affecting the cell behaviors across disease progression, right? So the first thing that we need to do for the spatial transcriptomics is to select the genes that we are interested in. This is not like a whole transcriptome-based spatial transcriptomics, but it can map up to like 5,000 genes here. And so the first step that our bioinformatics team did was trying to prioritize like similar concept of the random forest classifier again, but instead of prioritize perturbation, we prioritize genes that separating the conditions based on the cell type. That means we're selecting the genes that can really be identifying different cell types. And in the future, we are going to pick the genes from our single cell and RNA-seq analysis that is really glaucoma or like IOP relevant. So as a proof of concept, we first started with cell type marker genes, right? And then this is really work that's done by undergraduate students like in the lab. And then we were able to launch this like a random forest classifier really well, and then find out genes that are really important to separate, for example, RGCs rather than other cell types. And we designed a 100-gene retina cell type marker panel, including the major retina cell types and all the different like subtypes for different cells, including RGCs, amacrine cells, and bipolar cells. And this is really our very first spatial transcriptomics in the lab. And the machine was just installed thanks to the help from the department and Kirby Center. And it was literally installed last month, and postdoc Gabby in the lab did the first experiment. And we can load all the tissue sections on a single slide, and we're able to see this layer specific expression of different marker genes in each of the tissue sections. And she actually has a poster to talk about all the nuances for this technology. There's of course like more improvements to do, including like probe like optimization. But this is really exciting for us. And with this, we plan to repeat like the experiment and increase the data size. And hopefully we can use machine learning and AI to identify the cells and then the molecular signatures associated with glaucoma, so that we can further like screen drugs that can help with the therapeutic development. With that, I would like to thank all the people in my lab, especially Gabby, the postdoc, and then Jeff, the bioinformatician, who's really setting up the foundation for this work with help from Cass, Kelly, and Wei in the wet lab. And also many thanks to our collaborators, Chi, Mara, and Ken for their expertise in the glaucoma modeling. And thanks to the funding resources. I don't know if I still have time for any questions, but yeah, feel free to ask me.
I think we have time for a quick question while we get our next speaker loaded.
Curiously, some of the cells that were most perturbed seem to be different based on the different controls.
Mm-hmm.
Why do you think that occurred?
So we actually don't see much cell type perturbation by the if you're talking about vehicle versus the control groups, we actually don't see a lot of perturbations compared to like other comparisons like bead injections.
But I guess it's more about depending on which control you compare the beads to, by a slightly different order of perturbed cells eventually.
Yeah. So I guess like some cell types are still like more responsive to maybe like affected to some extent by the vehicle injections, and especially because we are combining. So this is like really preliminary. So we're combining 3-day, 7-day, 28 days. So we expect that some like cell types are going to be a little bit like stressed out by the vehicle injections anyway, especially in the earlier time points. So that's why we are, we have all the cells. We're just trying to increase the data size so we can analyze each time points like individually.